Thank you, Professor Straub. And we're going to dig a little deeper into science in the second session of the day and look specifically at dystrophin replacement therapies. Here to explain all about it is Professor Francesco Muntoni, who's the Professor of Paediatric Neurology and Honorary Consultant at the Paediatric Neurology Institution at University College London.
Good morning. I can see for the panic of your face that you thought, where is he? No, I was here despite the rain because we have enough water for the next century, I think. I was cycling. So, good morning, and thank you for inviting me. Believe it or not, it's a pleasure to be with many families with Duchenne. I wasn't here yesterday because, as Volker says, I'm involved in this European project of brain involvement in dystrophinopathies, and I was in Paris to discuss specific aspect of where the particular clinical part of the project is. And it was quite humbling to see the result of a large international survey that is going on with more than 600 families that responded worldwide, with the unmet brain need being approximately 50%. Recognize there is a major unmet need despite the age and approach. And I think the other thing that was there is the disparity of intervention. And I think these are all areas where we could do certainly better. We are not doing badly in the UK in many areas. This is an area we should really do better. Anyway, I'm not going to talk about brain. I'm going to, and I assume many of you know about dystrophin restoration therapy better than I do, and I will not go into the deeper detail of how an individual drug works and how another one works, because I would be surprised if most of you don't know more than I do. So it won't be a detailed assessment of this particular drug works in this way. There will be some general point, having said that. I have specifically avoided, and this is for a particular reason, to make any comparative analysis. The reason being that with the exception of drugs that have, as Volker mentioned, finish their pathway with a clinical trial, it is really very difficult. And I will warn you in terms of as investigators and you as a parent, how you should interpret data. What do data mean? And I will just say that I do have worked with a number of industry, as you expect. I will do some consultative work, mostly to try to help them to do a good trial. I don't have any shares, anything, and I don't get any money if a drug is sold or not. It's not really, my business is to try to help drugs to get to, good drugs to get to the end. So I will be talking about where we are with antisense oligonucleotides and with AAV gene therapies. Again, this will be a high level, and you're welcome to ask specific question if you want. Before going there, I would like to say a few general things that are important for you to think about when, for example, you talk about any of these drugs, but when you are asked to take part in a clinical trial, I think there are a lot of, if you like, a lot of issues to consider, and I think it will be important for you to question the colleagues that are discussing the result that are available, the issue about safety. I think that one aspect that has been important for the field in the last 10 years has been efficacy in targeting muscle cell. That will be a topic in my presentation that will be dealing with why the first drug wasn't fantastic, or actually why many of the first drugs were discontinued, and why there are many, many more drugs now under development. I think that you also should maybe consider and ask especially the frequency of the administration. And I mentioned that I have specifically avoided discussing, well, this drug appears to make 22%, this drug 15%, and therefore 22% is better than the other, is because there are major issues in the way quantification is done. And I think if an amica is here, Annemika. She would hopefully agree that, we co-authored this paper, that it's not so easy to quantify my dystrophin with the one in my lab, with the one from Annemika. Now, it would be easier if all companies agree to send it to Annemika. Then I think you will be able to say, one is 20%, the other is 18%, and there is a difference. But that's not the case, and you should always question. Well, and sometimes you won't have the answer. I don't have the answer as an investigator. What does this particular number mean? I think other thing for you to consider, and again, I'm talking from the family perspective, not from a physician perspective. When you recruit your children or you discuss about clinical trial, not all sponsors currently, for example, offer late phase home dosing. I think these are important questions for you. You may decide that it's not important for you. You may decide that it's important for you. But if you are living 6 hours from the center, maybe it is important for you. If you're living in the same town, it's not important for you. I'm only saying these are things for you to be aware in general and this will be important, of course, for chronic medication like antisense oligonucleotide. And I think that is increasingly most of the sponsors will offer on a compassionate use, unless there has been a safety issue, the drug to their children. I think it's important, after the clinical trial is finished, it's important to be aware of this. I like, when I start a discussion, to be clear myself of what may happen, because then I can explain, and then people can agree or disagree and say, well, I don't like this. Okay, but it's not my fault if the sponsor decided to do this or not. I have very limited aspects. So these are important aspects for you to consider. If I go back one slide, this is how the field is, and maybe there are one or two missing, but it is very busy, the field of antisense oligonucleotide. And again, I will not go back to explain how antisense work one by one, but essentially, one example here is if you have a mutation in this particular gene, in part of the gene, you can see that these two blocks don't stick well together. You can make them stick together by giving antisense oligonucleotide, deleting from the way the RNA is processed, this particular part, and then this block and this block stick well together. Of course, if you have a mutation here, well, you don't need to take this one out, so you need something else. And that is why there are multiple antisense that have been developed. They do not treat all people with Duchenne. They treat people with Duchenne with particular misspelling in that particular region. So I think the good news is that increasing, we started with actually some drugs that are not here anymore because they were eventually discontinued, but these are still in clinical trial now. So that we have, I would say, the most abundant clinical trial are the one on what we call the morpholino. There are a number of companies using them. The Sarepta is the more, if you like, the one that's been in the field for longer. But now NS Pharma also is pushing quite hard. I think that there are a number of modifications of this chemistry, the morpholino, that are where multiple companies are using different tricks to try to improve the amount of antisense that you need to put in the muscle cell. Because the antisense to work, they cannot work in your kidney. They need to go into your muscle. And in all these therapies, whether it's gene therapy, whether it's antisense oligonucleotide, whatever, really the aim is how can we preferentially target muscle. We have, people with Duchenne have less muscle than I have. I'm not particularly very well placed to say I've got a lot of muscle, but that will be nearly 30-40% of my body weight. And therefore, try to think how much is there, and therefore, that is where the drug needs to go. You cannot just not go to the muscle. So I think a lot of work is being done to understand, and I think this is probably also what Volker mentioned, how to build from what you learn. In actual fact, although it's painful, how to learn from the drug that didn't work. They were useful. I believe, I can tell you, those trials were not a waste of time. They were painful, but they were not a waste of time. You build on what, if you like, what didn't work, and how you move to the next step. And so all of these, especially the more recent drugs, try to build up in the understanding how you can better target muscle. And I think that if you like, the trick is that of, if this is the membrane of the muscle fibers, and this is this is inside, this is outside, where you want the antisense to go is really inside, where that particular thing where you want to take away, we want to trim the messenger RNA, this will be inside the nucleus. So for the antisense, our cells are done not to get stuff that we take that is in our blood. So our cells are quite clever in making sure not too many things get in. But little by little, I think that there is better understanding on how to, if you like, facilitate this process. I think it's always a trade-off, because you can make this process being very effective. If you exaggerate, there will be an issue about safety. So it's how you balance these two aspects. And otherwise, it would have been sorted 10 years ago. And this is moving forward, but I think that is where we are at the moment. So I have 1 or 2 slides per the different type of, and I chosen alphabetical order. I hope that I made I follow the path of alphabetical order of the companies that are involved. So they are not based on how far this program is or not in the development, but it's for you to get idea that there are multiple ways and multiple companies now in the field. So this is one that is called Avidity Bioscience. This is using, on the bottom of the slide, you will have how they are trying to do this. So they is one of the more recent generation antisense oligonucleotide. This is still very early trials. There is no final data release. They are doing multiple dose, ascending dose to see what is the safety of these doses. And then muscle biopsy will be one of the outcome of this clinical trial. And all these children then will go, whatever is the best tolerated dose, into an extension study to try to see. Also, initially this study will be relatively small, it will be open label, they will not have, they're not power to go to EMA and say, we have an effective drug, but they will allow to say, we think, I don't know, this is an example, 10 milligram or whatever, is safe, is well tolerated in the muscle biopsy of these children, makes sufficient dystrophin. We expect this to be effective. Now let's do a bigger trial. This is where they are. They started with healthy volunteer. They have identified what is the dose that they would like to take forward in the Duchenne studies. And I think they are, if you like, they have started to dose some of the boys with Duchenne. I don't even know, in all honesty, exactly where they are in this dose escalation. And we will hear quite soon whether this has been well tolerated in boys with Duchenne, whether there is dystrophin, how much dystrophin is there. I tell you again, be mindful. It's a bit difficult to compare exactly, but I think it is useful. I suspect it will all be with dystrophin, with hopefully all this program. But when you see a percentage, just think about that's a bit difficult. The second company in alphabetical order is BioMarin. BioMarin had been involved before in a clinical trial together with Prosensa. And that one of those trials did not work. This was at the very beginning, but I tell you, this was a good example of not having had success. Was painful. We were involved in that study, but allowed to understand how to do a better study. And that is what BioMarin now is doing. They are just about to start a clinical trial using a new antisense oligonucleotide that is much improved compared to what didn't work very well in the first time. And they are currently recruiting or currently opening sites in the UK. This will be a clinical trial for children aged between 4 and 10 years. that can walk, and who, of course, have not receiving other therapies at the moment, and in particular not receiving gene therapy. So, of course, there are no results of this in the human, but I think this is an opportunity, and I think, again, is building on previous negative experience. So that is always useful. This is another, alphabetical order, E, entrada. They're also using a system. They don't give enough information, so what exactly they're using, I cannot tell. But if you go into the website, they are using a way to link the antisense oligonucleotide to a peptide. They say it's a cyclic peptide. I'm not entirely sure what that means. Anemika will know it. And they are, if you like, already started a clinical trial skipping exon 44. We expect some early data this year, not in 20 years' time, this year, and not in a very long time. And then they have ambitions of they started with 44 to move to 45 and to 50 and potentially, or probably 51 as well as we move forward. So I think these are different ways to do it, and I think I cannot even believe that there are so many now moving into this. Now, this is maybe, hopefully, I didn't scramble the alphabetic order, but this is PepGen, so it's a P. And this is conceptually, it's not very different from the Entrada. The way it's done is very different, but conceptually is how I can take a naked antisense oligonucleotide, I stick something to it so that it gets into the muscle better. And that is what PepGen is doing. They have a exon 51 clinical trial underway now, and if you like, building on the experience. The good thing about this, if it works, it could work for other antisense. There is no reason to think that if it works for exon 51, it will not work for exon 44. So in a way, learning from one can translate to another antisense quite easily. And this is some of the clinical trials that are planned from PepGen, and these are if you like, where exactly these are. These are the name of the clinical trials. You will be able to get these slides from the UK afterwards. So don't necessarily take all the information, but at least it gives you a sense of which clinical trials are happening from which sponsor. So this is, no, I made a mistake with the order. No, as you realize, I'm quite self-conscious of being involved with some campaigns and the other. So I really thought, well, the only way is to make sure that I use a alphabetical order. So that's my poor English. So this is Dyne Therapeutics, should have been before PepGen. I think that this is yet another way, but it's slightly different from what has been used by the previous two, Entrada and PepGen, to stick a peptide. This time, yes, they also stick something, but they try to use specifically one particular receptor that is in the cell. The cells have got hundreds of small receptors. One is to get sugar in, one is to get things in. And this particular company is using one particular receptor to enter into the cell. Is actually, in actual fact, it's not fundamentally different as a concept as the first one, that is the avidity. So I didn't explain that particular one well. So as a concept, they're not very dissimilar. So Dyne is currently targeting exon 51, and they have already, let me see, what is the next one? Yes, so they have a dose escalation underway, and we should hear quite soon, maybe at Muscular Dystrophy Association in the States, or quite soon, what are how well it was tolerated, but also what is the dystrophin level in, I don't remember now, up to which of these cohorts. Some of these cohorts, the result will be available very shortly. Some of the other cohorts at higher doses, they will only be available later on in the year. But I think, again, this is just to let you understand this is all happening and will happen this year. I think that this is now NS Pharma, so it should have been Inashafat again before PepGen, but whatever. It was very late yesterday night on the train coming back from Paris. So, you know, so NS Pharma is a bit like Sarepta, is using the first generation NAC antisense oligonucleotide, and they have two clinical trials underway. The one for exon 51, they have reported some, if you like, short-term clinical benefit. They didn't show much difference, but they're using higher dose than Sarepta is using in most of the clinical trials. I'll come to that. And they showed, you know, potential clinical benefit in the short term. I think long-term data will be awaited. And I think more recently, they obtained, especially with a study done in Japan, a FDA conditional approval for an exon 44. I'm not aware the clinical data have been released there, so we'll expect some clinical data soon. And maybe we can discuss afterwards the differences in different regulators in accepting dystrophin as a surrogate endpoint or not. I think this is now Sarepta, and Sarepta has, if you like, two classes of molecules as a concept. One, very similar to what I said from NS Pharma, a naked antisense oligonucleotide, meaning antisense without anything sticking to it, okay? And they have several, etepilsen, golodirsen, and casimersen, but they also have, similar to PepGen as a concept, an antisense sensitivity, as opposed to Entrada as a concept, an antisense with sticked peptide to facilitate cell entry. The most advanced program are the one of etepilsen, golodirsen, and casimersen. The dose that is used in most of the trial is lower compared to the one in the two trials for NS Pharma, with the exception that there is a study underway, we are also involved, where significant higher dose of etepilsen are tested in people with Duchenne. So I think these are really studies underway now with closure that is not going to be, you know, in 20 years' time, but will be quite close. Because there are multiple things here, I summarize some of the main points that I understand. And again, I indicated I stay away from saying there is 22%, and I'm not making any comparative analysis. But of course, because these particular drugs have been for a bit longer, there is published data regarding some effect on the longer term, apart from the clinical trial, on loss of ambulation and lung function. I think one point that Volker made, most of these, and I think is mentioned here, most of these longer-term studies don't come from clinical trials because they come from long-term observational study. And maybe one thing that we could discuss afterwards, that I partially agree with what, I agree with what Volker mentioned, but at the same time, for these long-term studies, we need to have an agreement with regulator regarding what do they consider to be a valid long-term study. I think that, to me, is something that was not present. It wasn't completely transparent, both from the company and from EMA, regarding what they really wanted for ataluren on the longer term. And of course, the same will apply to many of these longer-term studies. So I think that so there is some open-label data that suggests both for etepilsen and less for golodirsen, because it's been more recently developed, that there may be clinical benefit. I think that on the whole, I think it will be fair to say these drugs have been quite well tolerated. These drugs need to be given every week, and that, I think, this clearly is a burden. I think important aspect is that there will be, finally, this year, some information regarding what is the real world in a prolonged clinical trial efficacy of two drugs, golodirsen and casimersen. That will be very useful to try to benchmark what is, in a well-controlled clinical trial, the effect of the first-generation antisense. I think this is, again, the second as I said, Sarepta had 2 class of compound. This is the second class of compound. This is the one where this naked antisense is attached to a peptide. And this is called the momentum study. This is a study underway. I think that it would be fair to say that while I've been cagey to say, well, this particular company or this particular drug makes more than the previous one, it would be probably fair to say that all the next generation drug make more dystrophin than the first generation drug. I don't think this will be disrespectful of any truth. It may also be true to say that the first generation drug are quite safe. So I think in a way, the important aspect is for all this next generation drug, will they be equally safe? Well, hopefully, yes. I'm not suggesting they may not, but I think because these have been used for less period of time, there is less information. So there is some information that has been released regarding the fact that this antisense next generation will make more dystrophin in the same laboratory setting. So this is a bit easier to understand because it's the same people who do compare 2 different drugs that they are developing. So this does certainly makes more antisense than the first generation antisense. I think that perhaps the only thing was that there was some adverse event in terms of some kidney aspect of hypomagnesemia. It is well controlled, but it needs to be monitored. I think this is something that not necessarily had been observed with the naked antisense oligonucleotide. A thread of all of this is that with time, people like me and many others, and the same is for all the antisense and for the AAV gene therapy, we are understanding that some of these drugs potentially may have side effects. It does not mean they are terrible side effects. It means we need to understand them, we need to prevent them. If you think about it, 20 years ago, none of us, as pediatric neurologists, knew how to use steroids. Or very few knew how to use really steroids. Now we probably are among the best people to use chronic high dose of corticosteroid. We understand what they do. We try to keep our kids as safe as possible because we understand what they do. So the fact that there may be adverse event, I don't want to say is a no-go. But for us, physician, we need to understand, and the sponsor, of course, and you, what could it be so that we are a step ahead. If you are a step ahead, this can be all managed. So now this is easy. W, I think is, I don't think there is a Z sponsor, so this must be my last one on the antisense if I haven't make another mistake. So this is, I think, again, an important concept. This is a slightly different concept of how the antisense are made. I'm not going to go there. I think, again, this is a company that failed an early clinical trial. We had, and other children in the UK were involved in this, but the company now really has built up on the experience. And of course, you think about it, they clearly don't want another failed trial. So I think that this next generation of this new development of this particular chemistry is, if you like, is in clinical trial now. There are several sites in the UK. I think I should have said that the DMD Hub provides an excellent directory of which are the sites that are recruiting your children. They also give information on how to express your interest for making sure that if you are interested, your children could be recruited. So I think this is enough for the antisense, because I don't think there is data here. There may have been some early data on dystrophin quantification that I skipped because of for what I said before, because it's not been done by Annemieke for all cases, okay? I will move into the AAV gene therapy. This is also going to be relatively high level. You are aware dystrophin gene is very large. The part in color is what you can squeeze in AAV. And it means you cannot squeeze the majority of it. The white does not. It's like in this room, we wanted 5,000 people. Well, I can tell you, it's impossible. At some point, you reach capacity. The AAV, with this color part, has reached capacity. It means, however, that some part cannot be there, but of course there has been understanding and development regarding which are the part of the gene that are less important for function. And we have known this also for Becker muscular dystrophy. We know which Becker muscular dystrophy do quite well. And of course, in time, this has been modified. There are, at the moment, a number of companies. There are the names on the left. I will give a brief overview where these companies are. They have chosen a broadly similar idea, and I think, again, you will see sponsors say, or this investigator say, this macrodystrophin is much better than the other one. It's not very easy to tell which one is much better from the MDX because they all work quite well in the mouse model. But there are some subtle differences, okay, in the way that individual sponsors have decided to cramp the room. So I'll start with, hopefully, alphabetical order. Geneton. Geneton is a charity with that is involved also with is a direct emanation from AFM. For this particular project, they are in partnership with Sarepta, and they are doing, and we are doing it. I'm the chief investigator of this international study. We are doing a clinical trial where, well, Geneton is doing a clinical trial where, at the moment, children have been treated with one dose, have been treated with a second dose, and very soon, in the coming months, there will be the decision whether the result of the second dose is good enough that remains the final dose or whether it needs to go to the higher, to yet another dose, okay? So we will know soon. This, when I say good enough, it won't be on the clinical ground, will be how much microdystrophin is expressed, okay? So this is recruited in London, in Newcastle, and in other European countries. Pfizer, together with Sarepta, other ones have done much more work because they started earlier and they are larger companies. And they have completed a study that is as a concept, not fundamentally different from what I mentioned to you from Geneton, meaning what are different doses we can give and identify an optimal dose for then doing a larger study. So this one has been completed. Then there are two studies that I will briefly mention that are in which the recruitment is completed, and we're waiting for the clinical data of these larger studies. So this is as I mentioned before, this was the study that was completed, and this was to find the optimal dose that could be used in the subsequent studies. Of course, all these children, because they cannot be redosed and you cannot take the AV away, they have been followed for the longer term to see also what happens in terms of the safety, but also potential efficacy, although this cohort is small. But the ongoing phase 3 trial, that is the one where, if you like, we will be and try to understand, or the sponsor will try to understand how efficacious this was. So this trial has stopped the recruitment, so this is a study that is underway. And the primary endpoint is to understand what is after 1 year, the difference between the children who receive the drug compared to the placebo, but then there is, if you like, additional milestone achievement that will be measured. So again, this is a study that this year should start to release some important data regarding the delta and the effect that this drug have had. And this is if you like, a smaller study. It's a phase 2 study. So it's not a placebo because this is looking at safety and tolerability in much younger children. We can discuss it later on, the pros and cons of targeting younger children. So the previous study was on ambulant children. This is still ambulant, but they need to be between 2 and 4. So I think, as you realize, powering a clinical trial for efficacy in this age group is very difficult. So the primary endpoint is can this be done? Is it safe? Is it tolerated better or worse than the others? And of course, longer term efficacy in terms of whatever these children achieve. Regent Bayer, hopefully again, RS, yes, it should be okay. So this is a clinical trial for yet another of the transgene. Is not fundamentally different from some of the other clinical trial or clinical trial design. Having said that, each of these have got some slight modification of perhaps some of the promoter that is used and where this isoform could be expressed. And I think this, again, is looking at the moment at which is the optimal cohort in a dose escalation of different doses and to decide which one is the dose to then take forward in a large study. So this study started to recruit last year. It's recruiting children between 4 and 11. I'm not aware of this being open in this country, but again, you will be able to find this information. And this is Roche, that, as you know, is the commercial partner of Sarepta. So this is what Roche will do in Europe, because we are in Europe, with the AAV developed originally by Sarepta. This is, if you like, where some of these trials are. I think that the EMBARK clinical trial, that is probably the phase 3 study, is the study that has concluded. This is the study that led in the States a conditional approval for a narrow indication. You are probably aware of this, so there will be additional data, information release, and eventually the decision of Roche or what to do at the European level regarding this. But there are other studies, apart from this one that is now completed. There are other studies that are on a non-ambulatory patient. These are all studies for which UK will be recruiting. There is another study, not very dissimilar as a concept to the one I mentioned before from Pfizer and the young one, but this one goes even down to 6 months. In actual fact, it says here 0, but my understanding is from 6 months onwards, but maybe I'm wrong. Anyway, it goes right down to very young infants. And then, of course, the long-term assessment of what is the efficacy of this drug. And I think this, I suppose this just summarizes with a little more detail what I just said. But if I go back here, EMBARK has concluded these 2 studies will be started to recruit for the different age ranges, older and much younger. And they will be recruiting now. So I think this states again at which stage these are and when they will be, when endpoint will be expected for some of the studies that will be started here. You have here the timeline, okay? These are my last 2 slides. I think similar to what I mentioned for the antisense oligonucleotide, we're also learning that AAV gene therapy is effective. It works very, is quite, if like the efficacy on some of the, especially surrogate endpoint, dystrophin, problem, macrodystrophin production, clearly is, these are drug effective. You don't have free lunch, there may be adverse event. I think that actually it's not that there may be, there are adverse events. To some extent, some of the adverse events is a bit expected. It's like to say, you give steroids, you hope there will never be weight gain. Well, sometimes children don't put on weight, but I don't need to explain to you that is not a given, and it requires you to look after those aspects. The same is for these drugs. They will have, and largely many of the adverse events are quite common. Some we are learning, and I think that is perhaps the other aspect for you to be aware. We learn how we move forward because we don't know what we don't know. And some of these new adverse events, for example, some of the adverse events regarding cardiac toxicity that has been noticed in some of the clinical trials, well, clearly it's not in the interest of anybody to have a severe cardiac adverse event. But if you never use that drug in that patient population, it's difficult to tell what may happen. The good news is that we are getting better in understanding what could happen. How can we monitor this properly, potentially which people maybe at the moment are at too high risk for the current generation to be exposed to this. So as a specific example, one particular adverse event that is an adverse event regarding an immune response against the transgene, meaning a kind of rejection that luckily occurs only for a very small number of children. This wasn't known before and occurred in multiple sponsors, multiple different trials. It's nobody's fault. Nobody anticipated. But then what you do is, in actual fact, this was probably a good example of the multiple sponsors coming together and agreeing what is why this is occurring and try to understand. Because I tell you, we always see the sponsor as having their own interest before anything. A sponsor doesn't need severe adverse event in the clinical trial, even just cynically. And apart from this, of course, you should see, many sponsors, the attachment to the clinical trial and to the patient is as strong as ours. So what I'm trying to say, this was not people pushing luck. This was knowledge gap. And we need to acknowledge that when we're pushing things in areas we don't know, you find the knowledge gap. I think in this particular case, together with, in actual fact, Annemika is here as well, but together with Carsten Bonneman and so on, we set up and all the sponsor an expert panel to try to understand better what's happening. This led to exclusion to a small number of children from the subsequent exposure, but for a reason, and people are working around this. But in the short term, it's better not to have severe adverse event. This was a particularly nasty one that you don't want. And then we hosted a European neuromuscular workshop to try to better understand this. I can tell you that I put all the things in the wrong alphabetical order apart from my poor English, because I submitted the night before, so this is Thursday night at 2 o'clock in the morning, another application to another EMC workshop because the deadline was yesterday. And because yesterday I was in a meeting, I had no choice. So this is a working group now to try to learn from ongoing work. And the MDA has done some fantastic workshop as well. The NIH has done another fantastic workshop. My message to you is we are all excited, and the same will be for the next generation antisense. There may be adverse event. I think we need to discuss them, understand them, getting better. I think we now are quite proficient for using AAV gene therapy in spinal muscular atrophy. We can learn, but I think we need to be prepared that some of these conditions or some of these novel therapies may bring things we didn't expect. So this is luckily my last line, luckily for you, my last line. So I think it would be fair to say that each year there is a step forward, not backwards, and not even steady state. When I was putting together these slides with all what's happening, it's almost overwhelming, and there are many, many things. I don't think the fact that there are competing programs is a bad thing. I think it's a fantastic issue. I think the other good news is that this year there will be large studies that meet the final endpoint. So we'll start to better understand what is the correlation between some of the dystrophin level and the clinical endpoint. This will be very good. I think I mentioned the safety before. And I think it will be fair to say, again, one has to finish, not necessarily on an optimistic note, but on a realistic note, that every year, when I put together the slide with all the antisense oligonucleotides competing, I was surprised about how many sponsors are now in the field. Maybe not all will work. maybe 1 or 2 or 3 will be better than the others. But, at least you have, if you have a single car racing, it's quite different compared if you have 20 cars racing. That gives a completely different perspective. So I think they are certainly exciting time. It doesn't mean there is a solution around the corner, but certainly a final solution around the corner. I think that this is the, well, I thank the sponsor, especially the Duchenne UK, the DMD Hub. We work very closely with them. I think in the UK, we are lucky to have also a pan-UK collaboration with the North Star. This is our team. This was my old boss. This is the away day we had last summer. This is the roof garden of our hospital. This is Viktor Dugovitz. I wish I learned more from him. Certainly, alphabetical order would have been important. And Carsten has been a close collaborator regarding the issue and the learning. And you need a colleague to trust that understand this to discuss very complicated things. And I think Carsten has been fantastic. So thank you for your attention. If you wish to hear my voice again, I'm happy to take questions.
Are there any questions?
I hope I didn't. I'm on time.
Hello. Hi. Thank you. For gene therapy, you talk about the AAV, which is, from my understanding, like a delivery method. Is there any research? So 2 questions, I think. Is there any research for alternative delivery methods other than AAV? And the second one is, I think, correct me if I'm wrong, my understanding is it's a single dose only. How's the research going for redosing?
Yeah, so in actual fact, you know what? I have another slide that I do not know. There should have been RS. So I swear they may be blocked, I may have made a mistake and is hidden from the presentation, but it will be a presentation on one particular sponsor. Again, is S, is that is solid. I not forgot, he's there, but he's not showing up for some reason. So I think that is addressed indirectly this, but it also allows me to correct the absence of solid. So solid is an example of one company that was, I have it here. It was there?
Yeah.
Again, scramble alphabetical order to confuse anybody. So solid is a good example as a concept similar to the antisense oligonucleotide. First generation really did not work particularly well. This was a standard AV9. And eventually the clinical trial was stopped, interrupted. Now Solid has done work on the next generation. This is the first of the AV gene therapy trial using a next generation AV. It's a modified AV. The concept is not fundamentally different from the second generation antisense, something that will target better the muscle cell. Of course, we'll still need to see whether this will be as safe, safer, or less safe than the old generation antisense. So they will start dosing, hopefully, next month in the States, and they're looking to expand in Europe and in the UK as well. So that, I think, indirectly addressed your question of what are these AV. Largely, what you've seen presented up to S is first generation AAV. Fundamentally, they don't work in a completely different way. I think will be, again, it's a bit like the antisense. It will not be honest to say this one works 100 times better than the other. There is hope that the next generation will allow dosing at lower levels, because that will be helpful to try to avoid some of the complications. For example, the liver complication is not infrequent in people dosed with the AAV. And so we will hear whether this one works better than the previous one. Regarding the redosing, I think at the moment this is a big problem. In a way with the current knowledge, the idea to redose a person is not possible unless there is something that needs to be done that has not been done yet. There are multiple ways one could do it in animal models. It's possible to do it in animal models that are naive to the disease, but in dystrophic animal models, it's been difficult to do redosing in dystrophic animal models. This is because there is already an inflammatory environment and so on and so forth. So it's not impossible in the longer term as a concept, but if you ask me today, I'm not aware anybody who has got a convincing protocol that would allow that to happen safely and efficaciously, because I think even if you do it again, the last thing you want is to spit out the second injection, if you like. So it needs to work. So I think at the moment that's difficult, but this will be. So we wanted, in actual fact, some of the advocacy group with whom we put together the EMC workshop program, we wanted that as one of the topics. We said this will require a 3 days full-time topic just on itself to give justice, because it's not just an hour, it's a lot of preclinical, clinical work. It really requires a lot of efforts. And at the moment, there is so much attention to make sure what we're doing now is safe that I think we start with where we are, but I think that will be one of the next one. Thank you.
Thank you, Professor Muntoni.